basically how, I mean, a lot of the guys have sent you big crop tests, uh, talk about the difference between fungi and bacteria,
Okay.
why it makes such a difference that we
Okay.
need to find that out.
Okay. I'm going to share a presentation. Is that all right?
Yeah.
I want to show some of. Can you guys see this okay?
Yeah, I see it now.
Okay, I'm not going to go through all the previous slides. I kind of want to start here, and then we'll talk about efficiencies and some of the stuff we're doing to looking at the way soil sampling has been traditionally looked at, and the way I'm looking at it now with more of a, I would say, adding another dimension by looking at genomics and total nutrients comparatively. So, good morning and afternoon, everyone. Thanks for joining me, and thank you, Joe and Terry and Kevin, for inviting me on the call.
You in your office, Al?
Can you hear me? Hello?
Yeah, I can hear you.
Okay. So, guys, I just kind of want to talk about generally what the soil sampling world has been and why I think we've got some issues in agriculture by looking at sampling as a model for giving us recommendations for fertility only. And just kind of show a little bit of the history of laboratory, the technologies that's involved in soil testing, and kind of where we're going at BTI and the way we look at stuff. And then give better, recommendations, and then also to improve, I'm going to call it efficiencies and functionality of the soil. How can we show you that by looking at a soil health calculation, if that soil can better get efficient use of nutrients versus one that doesn't have a good soil health calculation. So I'm going to show you that kind of study and kind of go through that. So traditionally, everybody in here probably knows what a Bray is when they look at a soil sample. They'll say, well, we want to look at Bray P. Dr. Bray and Kurtz came out with that termination back in the 1940s. Found out that pH had a lot to do with the available levels of P. So Olson came out and developed his method for looking at phosphorus in calcareous soils with his extraction in 1954. So these are two methods that now are still commonly used. Just imagine where we were with technology back in the 50s and back in the 40s and where we are today with technology. I mean, I like to use an analogy of where the telephone was back in the 50s versus today with cell phones and all that. We're still using methods that are really strong acids to extract these nutrients. Malik came out with his Malik 3 extraction in 1984, which was kind of the common method used today for looking at nutrients. None of this has been ever looked at what really is the function of the way plants transfer nutrients is through a digestive system, and these are all chemistry methods. So we're looking at a single extraction. 1984 is when that actually happened. Now you can still get what they call a saturated paste and use DTPA and ammonia acetate and that type of stuff now. But these are all strong acids that give you the numbers we're looking at. Now, what these numbers end up doing is they end up giving us, I would say, flawed data in the fact that we're putting out a lot more nutrients than the plant can actually pick up. And we have because of this over time, we have low efficiencies. Whitehead came out in 81 with the measurement to measure ammonia and nitrate. And that's kind of a lot of guys will send samples in, we won't even test for nitrogen. Rick Haney saw the problems with this testing back in the late 90s. And he began to develop what he called the H3 egg extract, which is it's 3 organic acids and water, which mimics the environment the plant's growing in. to be able to give us a single extraction. It'll give us a number that's based on not just chemistry and strong acids to extract these nutrients, but also give us more of an environment effect of what the plant is actually experiencing. So the H3A mimics the close acid that the plant exudates in. So the reason why Rick would say this is, and I believe it too, that's where I'm at, my history's in soil science, is that the environment of the plant is what we should be measuring, not these strong acids. If it was raining malic 3 acid every day or every time it rained, then we should use that extraction to measure our nutrients. But the plant's experiencing a different environment than what the lab data gives us. So that's what the Haney data is all about. And that really has went mainstream about, 13, 14 years ago. And that's why I like to use this Haney test to be able to actually give us really raw, serious data and what's available in the soil. So this kind of tells you where we are. We're looking at 10 million soil samples produced every year in North America, and we're using data sets that actually help us sell fertility, and mostly dry fertilizer. And I'm not here to just browbeat and beat down the dry fertilizer industry. I'm just saying that we're using a lot more nutrients in the form of inputs from fertilizers than we actually need to. There's no standardized testing method across the marketplace. You can use 4 different labs and come up with 4 different results. They'll be, generally the same, but when you're looking at what you're purchasing when you put fertilizer out, is your data solid, or is it helping the fertilizer company sell fertilizer? That's just my jaded view on it. And there really never has been a soil health test. So when I was working in the lab world, and I was instrumental in helping with the brand Waypoint, I really began to start thinking about what could we do to better get, information that would give us soil health calculations. So about I would say 2018, 2019, I ran across a group that's actually doing this method. And it's just one piece of what I want to look at when I start showing you what we're doing to give a better result when looking at a soil sample, is looking at genomics testing. So we know that soil is the gut for that plant. This is the holding place for the nutrition, not just nutrients, but other beneficial organisms that actually help the plant through a digestive system. Soils aren't just providing plants in the form of nutrition without any kind of interaction. They're doing it through what we call the rhizophagy cycle, or through a digestive system. And there is no standard method in the lab world that tests this digestive method of how nutrients are picked up. So the plant's the brain and the soil's the gut. Think of your soil like you would like a veterinarian would of the rumen of a cow. And that's kind of what we're doing when we look at why we want to better understand the way nutrients are picked up. So the traditional approach to soil testing is you look at your plant available NPK, you look at your pH, you look at your organic matter, we give a fertility recommendation, we put it in the spreader, and we go out there and we spread it, and then we hope, we base this on university studies that tell us that we want to get 230 bushel corn, we add 200 units of N. And these are all standard methods that have been used for years. And what we've ended up doing over time is building up these really high levels of nutrients in the soil that aren't being utilized. And we end up with these efficiency problems. I want to tell you now that the common thought process in agriculture shows us that the efficient use of nitrogen today is about 25% in low efficiency soil conditions to as much as 50%. So for the nitrogen you put down there, the actual amount of nitrogen going into that plant is about, for the most part, 50% of it. It's even worse when you look at P. We're looking at levels of around 15% actually utilized. This study here showed you that we got 33% nitrogen efficiency. So these researchers have pointed out inadequacy of the soil extraction methods. It's all across agriculture now. We know that lab data is giving us numbers, but the availability of these ingredients that we're putting in the soil are really not that efficient. And we need to find better ways, I think, as a general rule, to better get uptake of our income, what we're putting out there in the form of fertilizer and also the budget that it's costing to put these high-end products out there on acre of ground. Mulvaney came out with this study back in 2014, looking at the detrimental effects of potash on farm ground and what potassium chloride's doing, and what's happening when we apply these forms of KCl. We're learning that the efficiencies of those products are really low. We know that if a guy's going to use potash, he probably should put it out early in the fall so he can have available K when the crop begins to come out of the ground in the spring in the following year. It's a very, very inefficient form of K, and it's kind of a common product across the agriculture now. Millions of tons of this are sold every year. And what these products are doing, these high salts and these chlorides, are detrimentally affecting our bacteria and fungi in the soil. And that's really what's predominantly what's stopping these nutrients from being absorbed. So we want to know, here's another study looking at, this came out in 2015, showing the effect that MAP and DAP have on soils and what the low efficiencies are of those being. For most parts, farmers know that if they're going to apply a form of phosphate, that they're probably not going to get the benefit of that phosphorus for 2 years a lot of times. So this all led me to really kind of think about how we should be looking at soils and the amount of nutrients that are there. I also wanted to point out what we're beginning to really study and what is happening with these microbial colonies, because we know that the only way phosphorus goes into the plant is through the solution, right? It has to be into a liquid form. Any nutrient has to be in a liquid form or in a soluble form before a plant can pick it up. They don't have teeth. They can't chew these dry materials. These all have to go through a cycle to get the nutrients in the soil. And what we're finding out is the effect of glyphosate and glyphosate. I'm not saying don't use these products, but I'm saying these products have a tendency to break down the microbial, mostly fungal colonies in the soil. And that happens, we end up with these high levels of pythium, high levels of fusarium, high levels of rhizoctonia, and all these other pathogens. They could do better in conditions where we have high levels of glyphosate. So my rule of thumb is, if you're going to use glyphosate, be sure to inoculate. We need to bring these colonies back. This is part of that same study. We learned that field studies conducted in multiple sites have learned that soybean roots that are glyphosate resistant have higher levels of fusarium. And fusarium is a number. When I get these genomics tests back, I see fusarium levels that are off the chart sometimes in some of these farms. And when you end up with this pathogen dominant soil, that's just an example of why nature is trying to tell us that it's trying to balance and get these fungal colonies back. And the way that nature does that is through bringing in pathogens in the form of fungal pathogens that are trying to help balance this imbalanced condition. I just saw this in middle December of the importance of what's going on with, the Newsweek came out with an article saying that grocery prices are set to rise as more and more soils become unproductive. And what's causing this productive issue, and we're trying to build, get higher yields with fertility in management of nutrition, when we really ought to be thinking about how do we better improve our efficiencies by building up better levels of fungi and bacteria colonies that actually are going to be doing the digestion of these nutrients. Now, this just came out this morning. I put this in here looking at why we have this study is looking at soybean 6 nematodes and why we have now this really big issue with, it's affecting almost every farmer today with yield production on soybeans because of the high levels of nematodes that are there. And it's the same thing. Remember, the soil has two functions, really. We have fungal colonies that are helping us break down residues, helping us solubilize these nutrients, colonizing the roots. Bacteria solubilize the nutrients, and they move these through this digestive system. And what we've done with glyphosate and other things and salts and heavy fertility, heavy tillage, other practices, we've interfered with this process. And we end up with secondary problems like nematodes, for instance. They're attacking weak, stressed plants to try to build back these colonies of fungal colonies, mostly, that we've deteriorated over time through these practices we've been using. So we all know about the Liebig's law of the minimum. Liebig himself came out with this story that, you're limited, your yield potential is, basically limited to the length or the volume of nutrition in the soil. So if you have a low nutrient level, that's going to be your maximum yield based on the need of that nutrient. And we found out back in the 1800s when he developed this theory that nitrogen was a limiting factor. And when you took a really healthy soil and added nitrogen, you got this huge increase. Well, over time, we end up with these, and it didn't affect the digestive system as much when they first came out with it. Today, it's a little different story. Overuse of these inputs have caused us to be, with real heavy stress conditions. So he himself came out with this introduction to this story in 1843 from the University of Sciences, talking about how he realized how big of a mistake this theory was. And I like this little comment he makes here, talking about chemists and when they make mistakes in rating agriculture fertilizers, don't be too critical of these errors because he's based his conclusions upon facts which he can't know from his own experience, but rather has taken from agricultural text and beginning to believe that those may have been true and reliable. And I'm sorry to say, I'm a graduate from Oklahoma State, got my degree in soil science, took 20 some hours of chemistry. Fertility was the number one, I would say, topic in my conversations in my education. Took one course in biology, and it was plant biology. We didn't study soil biology at all, and this was in the 1980s. I don't even know if they're doing it yet in the universities. But I'm here to tell you, my 43 years in being in agriculture, that it is becoming very prominent that we have to start looking at how soils are actually functioning and how efficient are they in moving nutrients. So I call it soil metabolic health. How can we understand human metabolic health and what's going on with that today, because we know that for every time we take an antibiotic, we really ought to think about the effects that antibiotic has on our soil microbiome. Our gut microbiome. We need to think about the same thing and how when we look at adding inputs, what that's doing to the microbial colonies in the soil. So when we use conventional testing methods, we're looking at fertility requirements that can lead to really over excessive uses of fertilizers. And if we're going to improve these metabolic health, then we got to figure out what the functions are and how to better attack and put products out there that actually can address the functions. And when we then do that, then we can find an efficiency score, which I'll show you more of here in just a minute. And healthier soils require fewer fertility inputs. So when you see these high yields, they're usually in soils that are really functioning well. And in a soil's functioning well, you can get a better efficient use of the fertility. So what we got to do is begin to figure out what is it that we can do to help that soil function better. So our approach at BTI is we want to do this evidence-based decisions. We want to look at applications of inputs based on soil and plant testing. And then we can have better informed information, which leads to better inputs, right? We can better understand how nutrients are picked up by the plant. And so I call it a soil progress report. In this soil progress report, we want to look at the metabolic system. We want to know that functionality is a very important part of it. When I take a soil sample, the last thing I look at is the nutrient levels. I want to see what the abundance is. I rarely ever see deficiencies with total nutrients. I do see deficiencies when it comes to microbial active carbon levels, when I look at respiration, I look at, if a nutrient pathway is blocked or open, if the carbon cycle is working, that kind of thing. So we really want to understand what the soil solution is, what is the soluble nutrient levels, what is decomposition, and then what is the plant's appetite? Is it affected by stress? If stress is the dominating factor in that plant's environment, then it's going to be the dominating input that we have to do to manage stress. And that's what most all of our ag retail is all about, is managing stress with herbicides, fungicides, insecticides, fertility. These are all stress management. I call it stress management products. So the first thing we want to look at is, what is our soil biology? How is our digestive system working? Do we have nutrients that are going to become cycled? Is the metabolic pathways opened up? Are there good living organisms there? How can we have better microbial communities living in the soil and on the plant? The plant's essential to have these. That's the living energy to these microbial communities. The exudate from the plant is what keeps the microbial colonies alive. These aren't things you just add to the soil. It's the plant that keeps the function going. So just an example, on an acre of corn in June, the calories produced per day on that acre is equivalent to about 80,000 people calorie consumption. It's over 200 million calories per day per acre that's going into the soil through the plant's exudates. And so that amount of carbohydrates going into the soil are unbelievably valuable to what we're feeding. And if we don't have the microbial colonies there, then we have a stressed plant. And so that's what we're talking about with these metabolites: fatty acids, amino acids, lipids, sterols, alcohol, sugar, volatile organic compounds. These all contain essential plant nutrients. These are all, this is the way nutrients are carried through the plant, such as carbon. Carbon's a nutrient, it's a plant nutrient. Nitrogen, phosphorus, potassium, calcium, sulfur, all these are directly linked to the biochemical cycles that are driven by the soil microorganisms. So we wanted to really kind of, give it a solid view. I call it basic down. And I'll show you why I'm looking at this basic down approach. Is I want to look, the first thing I look at when I look at a soil sample is what is the fungi bacteria ratio. We'd like that to be in a really healthy functional soil to be 1-to-1 in a row crop. What is the carbon nitrogen ratio? I'd like that to be 10, 12, 11-to-1. Do we have stress adaptation happening in this soil? Or is the plant going to be able to tolerate stress? What is our carbon cycle? Are we generally building carbon? Are we burning carbon? Building carbon is an essential piece of relieving stress. Plants have the ability to take sunlight through photosynthesis and turn that into carbohydrates. So we know that having a better function in the soil, having better colonies of microbials gives us a better carbon story. So I look at the carbon story. Then I want to see what the disease pressure is. Is there going to be problems next year? If you took a soil sample now, looked at it for next year, what is the disease pathogens? What is the pressure we have? What is the stress management? And what's that going to cost? And then I look at nutrients. Are we going to be able to see what the total nutrients are, what the available nutrients are, and then are these nutrients going to be digested in this soil or not? We'll call this basic down, and our reports are basically designed to go through this model, through this process. So this is a before and after. So what I want to look at here is what, when we took a soil sample, what was the fungi to bacteria ratio here in this soil? And then after 2 or 3 years, we did another bee crop report, went from a 1 to 419 to a 1 to 3. And when you do that, when you begin to see that you can actually impact the fungal colonies by inoculants and adding better sources of, I would call it microbial active carbon, carbohydrates in the form of sugar, you can get better responses in the crop and you get a better, what I call a better function. The plant functions better. So I've showed this bottom piece here, the top part of our new functionality report, where I can take data from 3 different methods and then put that on one report showing the fungi to bacteria ratio here, what the respiration number is here, total carbon in the form of organic or total carbon, what is the organic carbon, and then is that pathway, percentage of pathway is open. In this case, 32% of that pathway is open, meaning that this is not functioning that well. It's a third as good as it could be. And so that's why balance is a piece of the first thing I look at. And also when I look at balance, I want to look at how mycorrhiza fungi is functioning. And when we talk about ectomycorrhiza and arbuscular mycorrhiza, Our products are designed to help the beginning stages of how plants are better adapting to their environment. So when you think about a veterinarian and you're thinking about soil in that same kind of process, when a baby calf is born, the most important thing that you can do is get that calf on that colostrum that the mother provides. We need to think about doing that the same way in farming. Seed treatment with mycorrhizal fungi and beneficial bacteria and colonies that actually help protect that seed when it sends out its first radical needs to be in an environment that's going to help it begin to start adapting to that stress that's going to be there. So mycorrhizal fungi are important to the function of that plant, and they're not all the same. Soybeans take different colonies than what corn does. So we've developed products that are more crop specific rather than just putting these ingredients in the soil in a broad spectrum. We know that soybeans need bradyrhizobium to help establish colonies that can help fix nitrogen. So we have to add bradyrhizobium colonies, different ones in our starter, our seed treatment, so we can get those colonies around that seed when it begins to germinate that can better colonize that plant throughout its life cycle and help it adapt to its environment better. So I call that a functional input. And then I want to be able to talk about an efficiency piece too with the carbon story. So the way that works, I'm breaking this down here. This is Dr. James White's diagram here from Rutgers that developed this concept behind the rhizophagy cycle, and the way these nutrients are provided to the plant through this cycle. So we talked about a lot of times. We talk about, and I came in on the call right when you guys were still talking about calcium. Is that this is the order of oxidative extraction from the nutrients through this cycle. First, if a plant could be in a stress condition, it's still going to live. It may be in stress, but the first nutrient that plant's going to utilize is carbon. It's beginning to build carbon through photosynthesis, and it's creating its cellular structure and its metabolism by carbon and oxygen. Third most important nutrient that we can really manage out there is manganese. Manganese is the oxidase form. It has so many functions. I don't want to go through them all here, but manganese is an essential nutrient, as is iron. Calcium's fourth place. Usually what happens if we have calcium deficiencies, and I can tell you that there's probably a blocked pathway. We have an issue with the digestive system absorbing that nutrient. Magnesium, copper, nitrogen is way down the list, zinc, phosphorus, and potassium. How many times have you taken a tissue sample with tons of potassium in the soil and you find potassium deficiencies in the tissues? It happens everywhere. And I see the ratios. We'll go, our cations looking at base saturation, and we think we have blocked, and calcium's not available because we have so much potassium's not available because we have so much calcium or manganese or magnesium. These are all issues related to digestion, not to available nutrients. So I look at the difference between how carbohydrates, using forms of microbial active carbon in the form of carbohydrates and sugar and humic acids, which help colonize these organisms. On the left, we have no colonization. We get very low efficient pickup of nutrients when you don't have good carbohydrates. And then the way carbohydrates work in solubilizing nutrients through the rhizophagy cycle, because these fungal colonies will build this mycelium and these networks of pathways for the bacteria to help solubilize these nutrients. You get a lot better uptake and more efficiency when you have good colonization of organisms in the soil. And then that's important to understand how carbon is cycled and how carbon is built and how organic matter is built over time. So carbohydrates are an essential piece of what we look at when looking at in the functionality of how soil nutrients are digested. So we got nutrients versus pathway. So I'm going to talk about the difference between a nutrient pathway and just nutrients alone. We know that nutrients are there. I can tell you now that I see levels of nutrients that are astounding when you think about how many thousands of pounds per acre there are of nitrogen, phosphorus, potassium, calcium, magnesium, iron, all of that. but we have very low available nutrients because they're not digested nutrients. So pathways are more important than just the nutrients. Nutrients are measured by chemistry. Pathways are measured by biology. Nutrients don't fix a pathway problem. So adding more doesn't fix it. And more food around the pathway alone doesn't do it. If you don't have the microbial colonies there, then that food source by itself, you're trying to grow colonies that aren't doing their function. So in my opinion, and it's why I'm working at BTI, we want to inoculate with a good source of carbon, a good carbon source. And then excessive nutrients lead to stress. It's the number one cause of stress. And then when we end up with all this stress, we end up with having to manage the stress, and everything we do to manage stress causes issues when it comes to nutritional pathways. So don't be somebody that wants to add more to get better results. That doesn't work. It's modes, molecules, traits, treatments alone. These all will not address the pathway performance issue. So I'm going to go through why we look at this in this way. So I work with Region Ag Lab. This is a lab that actually gives us the better insight in what we can do when we're looking at total nutrients, available nutrients, and then the nutrient pathway. All three of these different together on one report, different methods on one report. So here's a conventional total nutrient report. What we look at here is what the levels of total nutrients are from carbon all the way into zinc. I like to look at the carbon nitrogen ratio. And then on this side over here, if you were to ask for a total nutrient, you'll get an equivalency. Tell you that you have 5,500 pounds of nitrogen in the case. And you've got 12,000 pounds of urea. You know, here you have 500 pounds of phosphorus and 2,100 pounds of 1152O. In this case, over a ton, and this is a 0 to 8 inch sample of K, which is over 2 tons of potash. So the point being here is that I've rarely ever seen a nutritional deficiency when it comes to total nutrients. I usually see deficiencies when it comes to available nutrients. So in the same soil, that was the same example of that. You can see here that we had, and he just got through fertilizing in the spring, so this was a soil that was taken in May. 175 units of N. He's got 105 units or 105 pounds of P. That's in parts per million. That K is going to balance out to around 75. You can double that if you wanted to. Say you had 108 pounds of K. But the most important piece is, what is the life? What is the respiration, for instance? What is the amount of organic carbon? These are the numbers I look at first. In parts per million, I like to turn that into pounds per acre. What is the microbial active carbon percentage? And then what is the soil health calculation? So I use these indexes to form this new report I'm going to show you in a minute. Plus, I look at this functional piece. I want to know what the function is when we look at the ratio between fungi and bacteria. We're not looking at nutrients here in this column. We're looking at the pathways. What are the organisms that actually cycle carbon, help us build organic matter? Is the soil wet or is it breathing? What is the nitrogen cycle? You can't fix that with one organism. It takes multiple different organisms that actually can convert nitrogen to mineralized nitrogen, nitrate, ammonia, organic nitrogen. All has to go through a digestive system. So this is all telling me what the measurement is for that digestion. What is the phosphorus pathway? What are the cycling organisms that help phosphorus? What are the organisms that help K? And each of these nutrients. Now, these are pretty complicated reports. And so I wanted to build a report that can actually put it on one page. So then I can take each of those indexes and put it in a form that's better to understand. And so when you saw that report here, we were looking at total species. In this case, we had 443. In my report, I want to be able to show that up here at the top. And if we can improve it, then I should see an improvement of species over time. Go from 590 up to maybe 700, 800 after a year or two. If I can better I can see what my carbon nitrogen ratio is, what my microbial active carbon levels are, what's the organic matter, what's the pH, and then give you a total soil health calculation. In this case, we have a low number. If you did a sample, put a program in place, in another sample the following year, we'd like to see improvements. So I like to really kind of follow these and then put them side by side over time and then monitor that and see if we're better improving. What is my balance? What's my respiration? What's the hormone production, stress adaptation? What's the disease risk? Am I going to be susceptible to diseases? What are my biocontrols? What are the agents there that help naturally defend against pathogens, bacteria, nematodes, this type of stuff? These are all biocontrol agents. And then I'll take the total nutrients on the left of the sample, available nutrients here on the right, and then if the pathway is open in a percentage. And that one I was showing earlier is 32%. About half of that 90 pounds that we look at is actually going to be available because our pathway or function for cycling nitrogen is only 52% available. In this case, we're going to get better efficient use of P. We're going to get about half of the unit, half of the availability of K in this case. Calcium about half as well. 1,200 units available, only about half of that's going to be taken into the plant. So this helps me with an efficiency score. This year, I'm going to develop an efficiency meter or efficiency number. So I can take a soil health score and then show you what your efficiency is, and we can get better fertility management approaches. So I like to use our products with liquid fertilizers so we can get better uptake of nutrients along with digestion and inputs that help with the digestion of that. So I want to talk about phosphorus for a minute and how valuable this nutrient is and how we've overlooked a lot of the value that this particular macronutrient has. It's important to maintain adequate levels of available phosphorus. And I'm going to show you an example here in a minute of why we have a lot of problems because phosphorus becomes a really big issue when it comes to nutrient movement. It's a chelator. It helps almost every nutrient that goes into the plant is attached to a phosphate. And so it's a vital component to converting photosynthesis energy into carbohydrates. If we don't have the right levels of available P, and it doesn't take a lot, 0.1% of the plant's mass, but traditionally we end up putting it out there in ways that plants can't pick it up and we don't get it utilized. It's very low efficient input when we look at of how phosphorus is actually applied through MAP or DAP or other sources that we apply it. What I really want to focus on is why phosphorus should be looked at as in the form of a digestible nutrient. It's a key component in photosynthesis, energy transfer, cell division, cell enlargement. In my opinion, as a soil scientist, phosphorus management needs to be looked at probably more importantly than nitrogen management. Adequate phosphorus increases water use, improves efficiency of nitrogen, contributes to stress and disease resistance. We don't get better uptake of phosphorus by adding more dry phosphorus. So plant roots can only acquire phosphorus through a dissolved soil solution in the form of a phosphate. In inorganic or soluble phosphorus, it's very low concentrations in the soil, and I see that with the thousands of samples I look at over time. They're very low in the soil solution, and so it has to be replenished from the organic form, and it's only through a mineralized phosphorus that the soil microbial colonies, fungi, bacteria, can convert organic phosphorus into compounds that are inorganic, which is what the plant needs. So the rhizospheric bacteria, the ability to understand how bacteria and fungi play a major role in making these plant available nutrients, specifically phosphorus available. So I'm kind of on a soapbox talking about better understanding of phosphates and how cycling of this product or this nutrient is essential to helping that plant function. This is one of my friends out in western Kansas, and this is a yield monitor on his pivot on corn. And a few years ago, I would say 15 years ago, he decided to allow a pipeline company to come in and make a cut across that field and put a gas line in. And ever since then, he's had huge problems with yield coming off of that piece of soil. And I just wanted to use this as an example of why I think the soil functionality report helps us better understand what's happening here. So you can see that he had 238 pounds or 238 bushels down to the real dark red area of 0 bushels. And that's about a 90-acre swath through there. So I said, well, Cameron, let's take a sample of this area here where it's not cut. Give me a general composite sample of that area and give me a composite sample of the cut. And here they are side by side. So you can see that our soil health score, now he's been really focusing on this cut for a number of years, trying to build better levels of nutrient availability. But if you look at the phosphorus level, so you got a 30 soil health score there and a 13.5 where the cut is. Microbial active carbon is 78% in that the big area of the farm, that pivot. Respiration's way up, 274. And when you look at the respiration on the cut, it's 70. And when you look at the microbial active carbon, it's at 32. But what really shines is this phosphate story. It's got 1,400 pounds and only 4 pounds available. So now we can come in there and actually inoculate and good sources of carbohydrates. And we're doing the test this year to see if we can build that function back. And once we get these going, everything else will start going. So we'll have a story to report back to you. And I've done this on numerous other stories too, to see if we're better improving the soil health and getting better functionality. So this is our basic difference. I want to show you the difference between a heavily tilled, heavily farmed soil where they're going, Al, I really need help. We're not getting anything picked up. This is what I call we have two different reports. I have a basic report and a complete report. So in this case, I just took the basic test because I want to just look at the soil health, what the microbial active carbon levels are, what the respiration is. And I've developed this report to go from red to green. Red means bad, green means good. We all pretty much know that. Then I put it in the form of a gauge so you can see that where are we on the gauge versus, total nutrients versus available nutrients. You can see on this soil here, we have, if I was to just take a Malick report, it would tell me to add more fertilizer. Now this is a cut in a fence row, not even, 200 yards away. The difference between the soil there and the soil in the field is astoundingly different. I mean, the organic matter alone was 21% versus, 0.7. I don't see this very often. It's not usually this drastic, but the soil health calculation, if I would look at just taking a Malick 3 report, it would give me a fertility recommendation only to tell me my organic matter is huge, my pH is right in line. What I really want to look at is what the microbial active carbon levels are and what my respiration numbers are. So that's why we developed this functionality analysis. This is one that I want to just take a soil health calculation, looking at it with a complete report. I've shown you guys this earlier, looking at total species, soil health calculation. What is making me have a 7? And I can tell you here, all my pathways are blocked. Potassium's not bad, but my balance is out of whack. I mean, if I was looking at my respiration, I can see that that's, it's not breathing. This soil is suffocating. It's not performing well. This is somebody you guys know. This is one of Eric's farm, Eric Sweezy's location. We're doing some work with him now. We did soil functionality tests on most of his farm, and now I can tell you that he's building this soil health back. What he's doing is he's really showing that he's up at 660 species, microbial active carbons high, respiration is really high. His pathways are opening up. We've got a 30% carbons. We got to work on some carbon here. We got to get some inoculants in, but we're on a pathway to helping them have better efficiencies and higher yields. Well, I put this in there. I'm an Okie from Oklahoma, and this is where I grew up in the, my grandfather and people that grew up here knew all about the Dust Bowl and how soil conservation is an essential piece of why we built the Soil Conservation District and the importance of why we should manage better organic matter levels and, keep a living plant on that soil as much as possible and reduce that tillage if we can, but begin to help build soil health. This is one of our friends that is in Nebraska that began to start working with me about 10 years back, and we began to start really trying to look at how we could better get performance with his soils by adding better microbial colonies and improvement with getting colonies of fungal, getting better fungal colonies in the soil. So he sent me this picture last spring to show me the mushrooms that are growing as residue. This is a no-till farmer, and so it's really kind of cool to see that residue getting broken down by these colonies. And these are all available nutrients that are plant available nutrients. So these nutrients coming from a previous growing plant are readily available if they're dissolved and digested for the next crop. This is a nursery I'm working with in Kansas City, just inoculating. This is a red oak tree, the same fertility program, but just the difference between adding microbial colonies to that seedling when it's young as an acorn, and then growing that plant with the right kind of colonies on the root. So most of these nurseries that grow this kind of these plants in these conditions aren't growing them in soil. They're growing them in bark and man-made substrates that don't have microbial colonies. So adding these inoculants really change the effect of the way that plant grows. This is a black gum that was done the same way, non-treated, treated. This is pawpaw. We grow pawpaws in Oklahoma in the southeast, and the difference between just the inoculant of that plant versus the ones that aren't is phenomenal. Fertility doesn't do that by itself. This is a functional thing. Redbud. I'm throwing these in there just to show you that I like these examples because they're drastically different, because they're growing in these plants in these carbon-based substrates. So carbon alone isn't the total story. It's carbon along with the right kind of microbial colonies that can produce these differences. Treated and untreated. Root systems between treated and untreated with microbial colonies. So BTI, I'll talk a little bit about our product line. We don't just give out our organisms that are in the product. We're functional organisms. We like to look at the way our multi-species work in a system. We look at the cycle of the nutrient. We look at how those microbe colonies are specific for the crop. So we have specific colonies for corn and specific colonies for soybeans and other crops. We have them for rice and some others. And we don't have any single species products. Ours are all multi-species. High concentrations of effective microbes with really high colony forming units. So we are inoculants, and then we recommend applying carbohydrates with our inoculants. Think about it, from the time that seed's planted to the time it becomes a living adult plant, specifically in corn and beans, where you have that seeds laying in the soil there for quite a while, and if it's in a harsh environment, you usually don't get the benefit of the inoculant unless you have a good form of carbohydrate. So that's why I really am so intently looking at microbial active carbon in a soil sample. All of our ingredients are microbial and are encapsulated with a protein encapsulation, so we can mix our products in any tank mix. They're all liquids except for our talc. And the whole important role of this is we want to be able to mix them with fertility, with your herbicides. If you're doing any spray passes with fertility through the year, you can mix our ingredients with that without damaging the viability of the organism. So what separates us from the most is this protein encapsulation. Plus, we have this innovation process where every year and a half or so, we're always continually improving our genetic rate of gain. And then we also want to make sure that our colonies are the same. So we have a very intensive quality control program at our facility in Texas, to where we want to make sure when you get a jug of K-Phos, for instance, that those ingredients are the same every time you get it. It's kind of like, that's why I use that Coca-Cola example of the safe there. It's not the case with a lot of multi-strain microbial products out there. You're a victim of your environment when you're looking at compost teas. Now these are good products, but we want to make sure that our ingredients are always always the same at the same colonies, same populations. So that takes a heavy quality control method and system. We also want to look at data and driven approach. So we want to help the farmer by investing with that farmer to look at soil samples and then better place our products with that soil sample analysis. Well, these are our primers. This is our seed treatment products. We have a Prime Pro dicot and a Prime Pro monocot. And we also have products specifically for rice and other crops that we look at. This is our pit digester. When we look at, and I'm up in Iowa today as I'm talking to you, we have a lot of hog facilities up here that we want to be able to help better digest that manure, where we can add those ingredients back to the farm in the form of a spreaded benefit instead of something that's going to take away and just add nutrients alone. So a lot of these soils up here where they have facilities like this have really high levels of phosphorus. And so managing that is essential to helping us get better functionality in the soil. So we have developed this product called Devour. This is our nitrogen, sulfur, calcium product, carbon. It's more anionic in form. It's bacteria. 2/3 of the colonies in here are bacteria colonizing the plant root to help solubilize those cations. It has some effect on those anions. It has some effect on cations, but not like it does nitrogen. So this is more of our nitrogen product. It's a 1/3 fungi colonies with 2/3 bacterial. K-Phos was developed to help us better get utilization of phosphorus and potassium and other cations. Well, this one is really strong in the marketplace for helping us solubilize P. Just an example, for every ounce of K-Phos you apply in a furrow, you're going to get about 4 units of P and K. Unleash is a microbial product. It has microbial colonies in it, but it's also carbohydrates. So when we put our product in the spray tank, we want to have a food source for those organisms until the plant begins to take over and provide the carbohydrates for the inoculant. So that's what Unleash is. It's our sugar product. FinPro is our what we call biocontrol, biological fungicide that helps fight against those pathogens I was talking about earlier. We see the high levels of fusarium and Rhizoctonia pythium. This is a natural biological agents, multiple strains of beneficial organisms that help fight disease. Innovate is our stubble or residue breakdown product. Is a high fungal colonies. So if I see an imbalance with the fungi bacteria ratio that's really wide, and I just think we don't ever use enough fungal inputs because we do such damage to them. This is our product that helps solubilize those residues, help build higher levels of fungi in the soil. It's an energy form that we have it's a 90s trace minerals that help with stress. In a lot of cases, we'll get a new farmer and we'll have a lot of stress there. So we end up having to kind of put products in there to supplement until the soil becomes functional again. That's what this is. And then Cypro is our silica product. And, the role silica plays in functional soil is valuable. It's the most prominent element on the planet, but soluble silica is real tough to find if you don't have soils that aren't functioning right. So when you think about high nitrogen and what happens to that plant and that cell wall is it becomes weak. And so you end up with stress if you have a drought condition, that all that moisture and soluble nutrients that are in the plant go back into the soil. So Cypro helps keep that cell wall rigid and traps those nutrients and keeps them in the living plant instead of going back into the the soil. So this is another management stress adapting product we've got. It was a trial I did against Pivot back in 22. I just looking at the soil, mainly looking at the nitrogen levels of what, looking at Haney data of what the available nitrogen was in September of that year versus the levels of nitrogen from the Pivot Bio trial. This was a side-by-side activate trial I did looking at just input of using an inoculant without it, conventional corn treated, dryland corn treated with the same inputs, but the difference between using an inoculant versus non. This is a rice trial I did in Arkansas, looking at our Activate rice product with Defend Pro and Unleash. And in this case, I knew what the problem was. We're trying to see what the effects of potash were on this paddy rice and how we could better benefit getting uptake of P and K by using a microbial input. We used 32-ounce rate per acre and did 5 farms. And it's amazing, the 2 farmers that actually cooperated and left their K off got a 30 bushel increase that year. Now they're big users now. This was 2 years back. The ones that didn't really see much. So you hear a lot of that when you're looking at microbial inputs. So go, I used your stuff and it didn't work. Well, what are we doing to affect the function? So that's why I like to look at data. Sometimes our inputs alone affect the ability for those plants to function better. We just finished a tar spot trial with the University of Wisconsin, looking at the Defend Pro and tar spot. We saw this kind of summarizes, we went the different methods and what the amounts were we used. We saw a 350% increase on the non-treated versus the treated with Defend Pro. And we wanted to use Activate too in this trial just to see, because we know what those organisms are that help naturally defend against this airborne fungus. Had some good results with Activate too. Now, a lot of times we'll see improvements with yields using this soil applied nutrient or soil applied inoculant. Be honest with you, what we do is we see a real big reduction of pathogens, and that's the release we get with high. It's not just solubilizing the nutrient; it's also working as the plant's with the immune system. So we had a really good response from that one. This was some more of the data collected by looking at Defend Pro and Activate against the control with tar spot. Did a study with Texas A&M and a comparison. We're looking at Delaro with Defend Pro and Unleash, and we saw how our Defend Pro was actually stood toe to toe with that synthetic fungicide. There were some weather factors. Imagine that in South Texas, but anyway, that's basically my presentation. Hopefully, I didn't bore you. That's a little bit of what BTI is about and what we look at. We love to look at data. You know, Joe and I have been talking quite a bit about how we use our soil functionality analysis to better place our products. And anybody there that wants to work with us, I'm willing to help you out if I can. If I don't have the answer, I can certainly try to direct you to somebody that might.
So Al, help me understand your soil functionality test. Is that, or can that replace the B crop test?
No, it has the B crop test in it. It's one method. It's one piece of it.
I got you.
So I take a total nutrient, I take the Haney data, and I take the B crop off the same soil sample and make one report.
Oh, I see. Okay.
Because the B crop test alone doesn't give you any chemistry. It only gives you biology.
Yeah.
So I need to know what the levels of actual nutrients are. So, you know, you might have 11,000 pounds per acre of calcium and only have 100 pounds of soluble calcium, but your pathway may be completely blocked.
Mm-hmm.
And so just adding, you know, another form of carbohydrate doesn't do that. But if you could actually add an inoculant in there that would help open it up, I need to see that in a report. So I like to look at the totally available and then what's causing this imbalance. and it's usually a digestive issue. Almost every time it's a digestive issue.
Okay. So can you give us a 30,000 foot? What's the difference, or why do you see the difference you want to use Haney versus a Malik 3?
Because Malik 3 is a strong acid extraction. So Malik developed that he developed 3 in the 1980s. He started doing this in the 50s using a 2 pH acid extraction. And the reason they wanted to do this is they wanted to make a soil sample that they wanted to take an extraction that could go across all of the available nutrients. Now, they don't do it with nitrogen, so you could look at phosphorus, potassium, calcium, zinc, manganese, magnesium, all these nutrients by using a single extraction. And that makes that really cheap, right? You can get that sample down to $6 and $7 each. And so that was the driver behind what we call grid sampling. The problem is that it's only looking at available nutrients in the form of an extraction that's very acidic. So your soils aren't pH of 2. So what happens over time is we end up having really high levels of nutrients, right? You end up with these abundant levels. And so the Malik 3 was, and I'm an old Malik guy. I came out of that world. It's just not the environment the plant's in. It's not living in a 2 pH environment. So what Haney developed was a 6.5 pH environment with the H3A, right? And that extraction is more closely resembles the environment the plant's in. So that's the difference between Malik and Haney.
Okay. So Cassidy was on this morning. She mentioned a little bit about a PLSA test. How would you say that compares to taking a B crop, because one is a lot cheaper than the other?
Well, PLFA is measuring the phospholipid fatty acids. So fatty acids are every organic organism on the planet has fatty acids. There's different fatty acids though with fungi than bacteria. So what those phospholipids are measured with this method, it extracts those phospholipid fatty acids out of that soil to tell you what a ratio is between fungi and bacteria. The difference between a B crop and a PLFA is PLFA's measuring, the B crop's measuring the actual DNA. So we know that the DNA of fungi, and they got a library, so they'll take a sample, we'll measure the DNA. I need to plug in. We'll measure the DNA of the fungi, the DNA of the bacteria, and they're all, identified. 26, I think the library we're in when we're looking at is like 26 million, something like that. So now I can identify the actual organism, right? And then I can do a ratio of fungi and bacteria. That's the difference between a B crop and a PLFA.
Okay. And then there at the end, you did mention Defend Pro as a bio fungicide.
Yeah, these are, and if you've ever taken a B crop report, it'll say bio. My battery went dead on my laptop. But
okay.
If you take a B crop report, you'll see on there it says biocontrol agents. There's a piece on there that measures these. So we know that there's individual organisms that actually help defend against pathogens such as fusarium.
Mm-hmm.
And so we have those biocontrol agents in Defend Pro. That's what they are. So these are agents that actually help fight against pathogens.
Okay, so how much better is that versus the synthetic fungicides, and then also to address timing applications of Defend Pro?
So synthetic fungicides are not selective. They kill the pathogen and can prevent against pathogens, but they also do damage to the beneficials. And the beneficial fungi are the ones that we're trying to help right here. So that's why we developed Defend Pro. We want to be able to add these beneficial organisms that we sometimes kill and do kill when we use synthetics. So my whole approach is don't say I'm against synthetics, because they do work. But you remember years ago, and you would know it because you're a little bit older than me, we'd take
antibiotics. I mean, everybody used antibiotics 30 years ago, but we knew the problem with antibiotics was we ended up doing damage to our probiotics, to our beneficial organisms in our gut. So nowadays, a doctor may recommend an antibiotic, but they're going to also recommend that you use something to help reestablish and rebuild these colonies back. And Kevin, you probably know that in turkeys. I mean, a lot of these guys know they can't overuse antibiotics because what happens over time is you end up having problems with the animal not reacting to the antibiotic, and you end up with heavier pathogen problems. So that's the reason why I'm telling you, I'm not opposed to using synthetics, or I'm not opposed to fertilizers. I'm just saying if we use them, let's figure out how to better use them efficiently. And so if you're going to use them, then we got to remember we're doing damage to the microbial colonies, and those over time affect our health, our soil health.
Now, what about timing then on the FinPro?
The FinPro, we've done it as in the furrow and build those colonies early, but we're also looking at using them as better efficient uses of using them as foliars. So in the case of white mold on beans, probably, when you would apply your first fungicide, replace that with the FinPro, and then if you still see an infestation, usually I don't. It's really good on white mold. It's a product that we use broadly on white mold. But based on the soil health, if you have a really poor soil health, you're going to have a much tougher time defending against pathogens. So the healthier the soil, the more you can use a product like the FinPro and get better responses. But if I have guys using 2 applications of the FinPro and they're also having to use maybe an application of a synthetic. So, but we want to be able to begin to get you off of all the synthetic fungicides.
So,
Yeah.
Al, what is the cost on the FinPro for foliar?
It's equivalent to the price of a fungicide, maybe a little bit less.
Okay.
Yeah.
And
then-
it does, you've seen good response on that tar spot and southern rust?
Yes.
You done some southern rust? Okay.
Yeah.
So mentioning tar spot in your study, was there a different timing to try and overcome the tar spot?
I mean, you want to really think of this as a preventive, all right? That's our goal here, is because these organisms attack these stress plants. And plants go into stress every year. I mean, we're growing annual crops, so we know that.
Yeah.
But the level of stress is affected by the soil health. And so if we can get better response preventively, then you're going to have a better control. So usually, timing is important, and I can help you with that as we talk about how DefenPros improves the effectiveness of controlling those foliar diseases.
Yeah. You ever done any work on Goss's wilt?
Yeah, we have, but that's a bacterial infection. So
Okay.
it's not as functional. I mean, it has more of a bacterial functionality, but that goes directly. Usually when I see heavy levels of Goss's wilt, I'll see, and it all goes back to the gut, right? It all shows me that the soil's not functioning right.
Okay. Okay, you guys have any questions for Al?
Stay on this Defend Pro for a little bit. I mean, it kind of went with the idea that, post herbicide time, we were adding a low rate of fungicide at that time and seeing good control on like gray leaf spot early, and then helping later on with tar spot as well, not letting any other diseases get started in the plant. I mean, would Defend Pro at that time would probably be a better option, since you don't quite have the full leaf canopy, and you'd have fungicide hitting the ground at that point. I assume that'd be a lot better choice.
Yes, that's right. It would be, yeah, at that stage.
Would you need that if you did it in furrow instead, or
I'm
both?
going to tell you this. We're going to do this. This is adding a little bit of information, because our Time Pro, is we've started. We got one dealer working with us in Minnesota that we're actually adding the FinPro to his talc. So we think in furrow is probably, I would say 70% of the best effectiveness we're going to get with that, because you're going to grow those colonies with that plant all through the season as that plant grows. So it's an inoculant, just like the other organisms in our Prime Pro. So yes, I think in furrow is a great way to use it.
So if you're spraying it at tassel, how long residual do you think you get with it?
Oh gosh, it's nice. So we're not looking at it like an active ingredient, right? You're putting organisms into that plant.
Mhm.
These are all what we call endophytic organisms. It's the amount of stress. So I got guys that are doing it, one pass a year at 16, but majority of them that are really using it as a fungicide treatment are putting down two passes. So if you did it at tassel, I'd probably go with 4 weeks after.
Okay.
Okay, Al, as far as your biological, Defend Pro versus competition, is there a lot of difference between these? Or, you see yours being better, they're all about the same. I understand you're a salesman, so you like yours, but I just
I'm
want
a scientist. I'm a scientist. I'm a scientist first, Terry.
Okay.
I'm going to tell you this is when I was young, we never sprayed fungicides.
right.
And it's funny, whenever Roundup beans came out, we used to see rust on beans everywhere before. All of a sudden glyphosate stopped all this rust. So I'm of the opinion that, like I said all along, these modes of action work. We know that the plant these organisms build a resistance against them, right? Over time. That's why some of the Syngenta products have 3 different molecules in the product now. What they're doing to the soil biology is what scares me.
Right.
Is we end up deteriorating our functions, and we end up deteriorating our levels of beneficial fungi with all these fungicides. So all I'm trying to say is that if we're going to use these products, we need to inoculate with good organisms that help build these functions back. And we're not going to do that in just one time. One 8-ounce rate of Defend Pro is not going to fix it, but we can begin to help the plant defend itself against the pathogen. Which anytime we see a disease, that's a red flag that we have stress.